All right. Hey guys. So this is going to be the lab mini lecture on the genetics of PTC tasting. And I'm going to try and keep it as a mini lecture, but the nature of this lab, it's pretty complex, a lot of molecular biology concepts, and it requires a good amount of explanation, and I'm going to try and do that to the best of my abilities, make it as clear and effective for you guys as I can, and try not to keep it, or make it so long of a lecture, and really, get to the points, the very critical, important points here, for your understanding and for your ability to complete the lab. The first thing I'll say is 2-week lab, meaning you have 2 weeks to finish the lab. This is because part of the lab, there's a procedure called PCR, and it takes like 4 hours. So if we did this in person, you would start PCR, but we'd meet the next week to finish the lab. But obviously we're not doing PCR in person. So I'm just giving you 2 weeks to complete the lab, which I think is great because, again, it's pretty, hardcore. It's a lot of in-depth material here, and I think it's good in that way. It exposes you all to what molecular biologists are doing all the time in labs all over the world. These are these lab techniques that we're going to be talking about are extremely relevant and important. So without further ado, let's begin. Actually, before I get into the concepts here and what we're going to be talking about and doing, not only do you have 2 weeks, but all there is to complete is the pre-lab questions. So here are the pre-lab questions like you normally do, and the post-lab questions. Really, that's it. There's no other things to complete. Yeah. So pre-lab questions, post-lab questions. That's what's due. There is a tiny little fill in the blank here for strength of flavor. You taste something, like how bitter is it? You can put any number or no number, since we're not actually in person tasting the compound. Okay, so let's begin. And I'm going to start here by introducing what this lab's about. And there's this chemical, PTC, has a very long, complicated name, phenylthiocarbamide. And it's a bitter substance, and about 70% of people in the human population can taste this bitterness. I believe I'm a taster. And so there's these PTC strips that have the compound on it, and you touch it to your tongue, and you can taste like this really kind of terrible tasting bitter flavor. But then, of course, 30% of people can't taste the bitter flavor of the PTC. And it turns out that your ability to taste the flavor or not is determined by dominant recessive relationship, which we just finished. Let's see. Okay, I am back. I had to look at our syllabus and kind of see where we're at. And there are some things in this lab that'll be a lot more clear after our lecture Thursday, but giving you 2 weeks and introducing these ideas now, a lot of which we have discussed, with homologous chromosomes and things like that. I think it'll be good. I think this is a good place to introduce some new things that we're going to discuss further on Thursday. Okay, because I was debating delaying the lab until next week, but I think introducing it now and getting it to you now might be actually better. Okay. As I was saying, this PTC, right? This compound, this chemical, 70% of the population can taste the chemical, 30% roughly are non-tasters. They cannot taste the bitter compound, the PTC. Okay, so there's a gene located on chromosome 7 that encodes for the ability to taste. It's actually a gene that determines a protein. So this is something we've alluded to, that the DNA stores our genetic information. The primary understanding here that we're going to explore a lot in this next unit, because you're taking your exam right now, at least my synchronous classes, is this central dogma of biology. And it goes like this. It's called the central dogma because it's so foundational and so important to biology that that's better. This is the first sort of understanding I want you to have. So DNA stores, it is the template, right, for our genetic information, meaning our characteristics, our physical characteristics. And DNA ultimately codes for, it is the recipe for, I think we talked about sort of our chromosomes, DNA being this encyclopedia. It's information, right, that our cells read in order to produce proteins. This process of how this occurs, we're going to explore in depth during this unit. So it will relate to this lab. You know, I'm pretty new to Mira Costa, so it didn't line up as well as I had hoped. But I think introducing it now and then kind of solidifying it, I hope, during the lectures will be useful for everyone. In fact, maybe it'll work even better to have it in your head now and get some sort of understanding now and then a more concrete understanding later, whatever. Okay, so that's the idea. We use our DNA, we read the DNA to produce proteins. Specific regions of DNA in our chromosomes are going to encode for different proteins. We've talked about proteins at length, proteins like antibodies for host defense against viruses and bacteria. We've talked about structural proteins in our skin cells. We've talked about enzymes, of course. These are proteins all encoded for in our DNA. Okay. All the instructions to create these proteins are in our DNA. Now, back to this PTC. So 70% of people have the DNA sequence, the gene sequence specific for this protein, a receptor protein, a taste receptor to be specific, that is expressed or seen or produced in the taste buds. And so this is going to allow for some individuals who have this gene or the version of this gene to taste bitter things, such as the lab says, Oh, well, we have about 30 different bitter taste receptors. This is one of them. And this plays a role in how we taste dark chocolate and coffee, cabbages, Brussels sprouts. Uh, so essentially, we've discussed evolution, and we as humans have evolved this ability to taste bitter compounds, mostly because poisonous plants that would kill sort of guided us to evolve with them and recognize these and spit it out or not eat any more of it, right? Okay. So, quick recap, the 70% of people have the version of this gene. So we did talk about homologous chromosomes, right? Having the same gene at the same locations, but you can have different versions of these genes, like hair color we've discussed. And the different versions of this gene there's two. You can be a taster version. So the DNA encodes for the receptor, this protein that in your taste buds that allows you to taste this bitter flavor, or the DNA is a different version. The DNA sequence is different, and it actually encodes for a faulty receptor that doesn't work, and you can't taste the bitter compound. So we have two versions that we're working with in our DNA, and therefore really two proteins that we're talking about. Okay. So back to our DNA here. There's a thing called a SNP, SNP, and it stands for single nucleotide polymorphism. And all of that fancy jargon, all it means is a SNP. Now, keep in mind, in our cells, our DNA, our genome, the genome is the entirety of DNA that we have in one of our cells. It's about 3 billion nucleotides long. A SNP, single nucleotide polymorphism, is when at least 1%, so in that genome, 3 billion nucleotides, if there's one of those nucleotides that differs in at least 1% of people, we consider that a single nucleotide polymorphism, or SNP, SNP. So if there's more than 1% of people that have a different nucleotide at that position, remember there's 4 in DNA, you can have adenine, thymine, cytosine, or guanine. We consider that a SNP. And let's see, so okay. Interestingly enough, so let me preface this, every human doesn't have the exact same DNA, right? That's why we all look different, act different, etc. It's very similar though, very similar. I think every human has about like 99.9% similarity in DNA. But there are these SNPs that occur throughout these 3 billion nucleotides. And okay, so just see here. So like I said, we're talking about 2 versions of this gene. When we discuss genetics soon, these versions, we call them alleles. So you can have 2 different alleles for a gene. And you can either have a dominant allele or a recessive allele. And so the dominant, so let me write that actually as well. Dominant or recessive. 2 different versions. Okay, the dominant allele in this case is the taster allele. And the recessive allele is the non-taster allele. Okay, now let me show you, actually write that as well. So this is taster, this is non-taster. Okay, let me show you, I'll be sending this out to you guys. What we call a sequence alignment, where we compare two different DNA sequences side by side. And essentially they aligned someone with the taster gene version, so dominant, and then someone with the non-taster. And so they align it like this. Okay. Now, I have some of my notes here in the pen, but you can see these three arrows. Okay. So all of these letters are the exact same between these two people. Exact same. And like I said, 99.9% similar, right? But at those three arrow locations, you'll have this in front of you, there's a different one. There's a different letter, and these are 3 SNPs. So in the population, at least 1% of people, this is different in people. Okay. For each one of these positions, there's 3 here. Now, they're different in this gene. So the non-taster version, so this is important. So the non-taster allele, if you have a non-taster allele, it is not highlighted. It's not the dark gray one. It's just the one that's the top. And then the dark gray highlighted version, that's taster. So they have the SNPs that are listed that are highlighted. Okay. Next concept. Here's what is going on. In this lab, you use what are called restriction enzymes. You use a restriction enzyme called FNU4H1. Okay, and what a restriction enzyme does is it cuts DNA. It cuts it. DNA is double-stranded. It cuts right through both strands. Boom, chops it in half. Kind of like if you pretend this is my double-stranded DNA here. It just cuts it right in half. Demonstrate that. Here's my DNA. A restriction enzyme, what it does, it just comes right in and chops it in half. Okay. Now, okay, so a restriction enzyme will only do that if there is a certain restriction site present. And the restriction enzyme FNU4H1 has or recognizes the recognition site that, so this is actually another fill in the blank you can put. It recognizes here. So again, double-stranded. So this restriction enzyme is going to see this in the double-stranded DNA, and it'll cut there. It'll make a cut. And the only sequence that has that specific site is the taster. I'm just double checking if I looked carefully. Okay, yeah. Okay. Yes, perfect. Okay, so the taster. So you'll notice the taster person has that recognition site. Therefore, if you're a taster, that restriction enzyme will cut your DNA there. If you're not a taster, you don't have that specific recognition site, and the restriction enzyme can't cut there. It won't recognize it. Okay. So what you're going to do in this lab is get your own DNA, extract it from your cells, and apply this restriction enzyme and see if it cuts, because that will determine if you have a taster allele or non-taster allele. Okay. Next page here. Okay. So, like I said, the genome is 3 billion base pairs long, and there's plenty of these restriction sites along with that, right? You're going to have that specific GCN. GCN just stands for any nucleotide can be there. You're going to have that along the length of that restriction enzyme is just going to chop a bunch, and then we have nothing to work with because we can't tell if it chopped that gene or not, right? So we have to look at this gene specifically. And the way we do this is PCR. PCR is this molecular biology technique. It stands for polymerase chain reaction. And this, I'm going to pull up some slides to show you what we do. Okay, so the first thing is PCR allows us to isolate a specific area of that massive genome. So isolate it, and then it also allows what the purpose of PCR, the main purpose, it's a chain reaction and it amplifies DNA. So if you have even a tiny, tiny, tiny amount of DNA from blood sample, from hair, from skin cell, that's what we're going to look at in this lab. You can make tons of that DNA to work with. Otherwise, you don't have enough to visualize. We're going to be visualizing DNA here in this lab after we make a bunch of it. And of course, it's tiny molecules, so we need a lot. Okay. So the best way to explain the PCR process, I think, is to look at this slide here. I'm going to send these slides to you for additional clarifications and such. Okay, so in PCR, we have our DNA. So DNA, we're actually going to extract in this lab. So I'll briefly talk about that. So the protocols in this lab for completing all of this is, it's a lot. And so I'm not going to really go into that very much. I just want to keep this at the concept level here. So you're going to have the DNA from actually our cheek cells that we're going to use. we're going to have what we call primers, which I'll explain in a moment. Nucleotides buffer to keep the polymerase stable. And yes, this polymerase, it's called Taq polymerase. Okay. Okay. Just looking at my notes. So here's now that we're talking about this polymerase ACE ending, right? It's an enzyme. It's Taq polymerase. Taq stands for Thermus aquaticus. It is a bacteria that lives in extremely hot environments. They actually first isolated it in Yellowstone hot springs. So these bacteria live where it's boiling water temperatures. So essentially, this is a heat stable enzyme. Remember when we learned about enzymes that under these hot boiling conditions, they're going to denature. They're not going to work. They're going to unravel and lose their shape and therefore their function. Well, this enzyme functions at high temperature because that's where these bacteria live. And it's a DNA polymerase. As we're going to see, to make all this DNA, we need to make more DNA, and it's going to make more DNA polymers. Now I'm going to show you how on that slide. What was I going to say? The bacteria, the hosmine. Just that it was a brilliant discovery. This researcher cleverly said, PCR requires us to boil our sample. We got to heat it, and I'll show you why in a moment. So we need this heat stable polymerase, or else this won't work. And so that was a really clever thing that this researcher did, who invented PCR. Now, if we look again here, let me explain what PCR does. So just in theory, you only need one DNA strand, just one. And so we'll take a look here. The very first step of PCR is called the denaturing step, where you boil essentially your DNA, 95 degrees Celsius. So the temperature that you boil your eggs or pasta, it's like 100 degrees Celsius. It's extremely hot. It would kill you, boil your skin. You don't want to put your hand in there, and denature your enzymes, but not this bacteria. So it's pretty cool. But the reason you do it is because during the denaturing step, it separates your DNA. It unwinds it. It opens it up like this. Okay, this double-stranded DNA. And the next step is called the annealing step. We cool the sample down so that we can have these primers bind to either side of the DNA. Now, this is where the selective sort of process comes in, where we're not amplifying the entire 3 billion base pair genome. We're just amplifying the region of interest. So the primers we supply are specific to the taste, or to this gene, this taster non-taster gene. And so it's going to bind there to either side. And importantly, our third step, the extension step, this is when Taq polymerase comes in and extends out. You see the arrows here. Adds in nucleotides that we also add to create, at the end of the day, 2 brand new identical DNA copies here. 2 new double-stranded DNA molecules. And again, let me just mention, the polymerase only recognizes pre-existing, it can only extend out from double-stranded DNA. So it can't just bind anywhere. It only recognizes where these primers are and extends out there. Exactly. And our primers, like I said, we design them, we order them from a company, and we get them specific to the gene. So the only place you're amplifying is the gene of interest. So it's pretty good. Now, this is a thermal cycler. This is what you do PCR in. Extremely expensive, but Mira Costa has one, so it's pretty cool. And it cycles, right? So we went from hot, we cooled down, and we go a little hotter so that the Taq polymerase is at its optimum temperature and it's functioning really well. And then at the end of the day, we get 2, right? Well, it's a chain reaction. So this is why PCR takes 4 hours. How long has it been? Okay, I'm doing okay. Might go a little over, but that was planned. It's as you can see, this is pretty crazy stuff. Okay. Oh, 4 hours. It's a thermal cycler. It's not going to just do this once. Here's one cycle, right? It's going to do it a lot. It can go up to like 35 cycles. Check this out. You start with one strand of DNA. Here's our gene, the wanted gene, or our gene of interest, the taster gene. And after one, as we saw, we make 2 brand new copies. Then each one of those is going to undergo the same 3 steps: the denaturation, the annealing, the extension. And then you end up with 4, and then 8, and then 16. Then if you do 35, you get 34 billion copies of DNA. This is actually how they get DNA at a crime scene. Crime labs at police stations, they do PCR all day. They get it. They only need a little bit of DNA, then you can get a ton, and you can sequence it and identify who it is, who left the blood there, or who was at the scene, whatever. So we want a bunch of DNA so we can work with it. Here's some videos if you want additional clarification as to what's going on. I'm going to send you these slides, like I said. Oh, the protocol, don't worry about this. Of course, like I said, we're not in person, and it's irrelevant for an online lab. Okay. And okay, so these are the 2 spots where the primers will go. And so we're going to amplify the region that contains the recognition site. Now, I mentioned the recognition site is at this SNP. There's 3 that are different between the taster and non-taster versions or alleles. But the recognition site, the enzyme we use to cut the DNA, this is the important one, and we're going to amplify it. Okay, the gene at this region. So this cut in the DNA, that's what we're going to be looking at. Or if it doesn't cut, PCR product restriction enzyme. So I mentioned FNU4H1 is going to digest the DNA or cut it at this site into 2, like this. And boom, you separate it out like that. And like I said, it's only going to cut if you have the taster sequence, that C there. And the non-taster has the T, so you don't get it cut. So we apply that. Now, I haven't mentioned yet how we get our DNA. Just be really quick about that. But that's the first thing we need to do, right? Before we work with DNA, we need to obtain our DNA. And in this lab, we get it from cheek cells. And so you swish with salt water and spit it back out. You get some cheek cells in there. And then there's this whole section on obtaining cheek cell DNA. I won't go in depth here. But essentially, we learn that DNA in our cells is in the nucleus. And if we have our cells, we want to release the DNA. We want to work with the DNA only. So you got to lyse your cells, extract the DNA, and isolate it. And so those are the steps to do that. You can read it if you're interested. It involves using these Kellex beads to prevent DNA degradation and these things. Okay. And if it sounds fun, then you'll love biotech and research. There's a lot of pipetting, but okay. So once you have your DNA out of your cells, your cheek cells, you can do the PCR that we just looked at and amplify that gene of interest. Once you amplify your gene and you have a ton of copies of your gene, DNA, you can then, and so this would be like week 2, you'd come in and we would apply the restriction enzymes to your amplified DNA. Okay, I mean, that's the heart of this lab. There's some stuff I want to talk about with, well, actually, one more thing, and then some stuff with the results that you get and how to interpret them, because that's what the PostLab questions are really about. Okay, so first, let me start with PCR. You always do it with DNA gel electrophoresis. It's called gel electrophoresis. In fact, I think that PowerPoint slide has, oh, what's going on? Okay, I think this is working. Oh, okay, it's showing us, oh, this is good. This is showing us after you add the restriction enzyme, if you have the taster version, it'll recognize the recognition site with that C there, that SNP, that's a C, and cut the DNA. Doesn't recognize if there's a T there, and so FNU41, after adding it to the mixture, will not cut your amplified DNA at all. Okay, so let's take a quick look here at gel electrophoresis and kind of the concept, the principle of that. So once we have our amplified DNA, and really you do this whenever you do PCR, if you want to analyze DNA, right? And then we cut our DNA, our amplified gene or not, if it's the non-taster version, right? We run it on a gel. And it's an agarose gel, it's just type of sugar. And you run a current through it. Now, the important thing with gel electrophoresis is that DNA migrates through the gel. DNA travels through the gel. This is because DNA is negatively charged. It's a negatively charged molecule. And this power source has a negative end and a positive end. So the negative end, the DNA is attracted to the positive end, and it's going to migrate through towards the positive end. Okay. So you have different samples, basically, you really would have one with your DNA in it and see what the banding pattern would look like. But sometimes, let's say 20 students ran on one gel, then you'd have 20 lanes, you'd have 20 samples running. And this is the kind of thing on the right side here that you would see as a result. Importantly, these are DNA strands that were amplified. And based on how large the DNA is, so say you amplified a massive gene, you have a ton of DNA, but it's all really big DNA. All that DNA travels very slowly through the gel. So the larger the DNA is, the slower it travels. And then in contrast, smaller DNA travels faster. The analogy I like to use is kind of like a baseball, throwing a baseball versus throwing a bowling ball. If it's larger, it's not going to move as far. Smaller, it moves really quick. And so this is a smaller fragment of DNA. This is even smaller than that. We're assuming they applied all this at the same time and ran it for like 30 minutes. So the largest fragment of DNA is up here, right? And this is also pretty big. Now, why would that matter? Why would a banding pattern matter? If you're thinking about this kind of percolating in your head and we're cutting DNA, having two smaller pieces versus larger, right? Then maybe this is coming together a little bit. So at this time, I want to discuss what I'm going to send you for the PostLab questions and data that I want you to interpret. And then graph a set of class data that was made up. Or maybe I think Stacy got it from a previous semester. But importantly, I want you to know how to interpret these gel results, what they mean. And it does touch upon a concept we have discussed with homologous chromosomes. And let's take a look at the data here. Oops. So I definitely think that answering this, after our genetics lecture, it's going to be a lot more clear. But I think introducing it now is not a bad idea. Come on, share. Okay, so these are 3 actual gels that I'm going to send to you guys as part of the PostLab. And you're going to analyze these results. And you're going to tell me what, I think my roommate's cooking. I'm going to tell you what, well, I'm going to tell you what each of these means. Okay, so let me go ahead and do that. It has to do with homologous chromosomes, and it's referred to in the lab as genotype and phenotype. And you're going to determine student 1 and 2 and 3, you're going to determine whether they're tasters or not based on their gel results, based on if the restriction enzyme cut or not. As we saw, the restriction enzyme cuts when they're a taster. So I want to paint this picture for you very clearly because it can be, looking at that, it's a little abstract. So let me show you. Each one of these students, students 1, student 2, and student 3, remember I mentioned in the beginning, I hope, this gene is located on chromosome 7. I don't know why I put a 1, 7. This taster gene for PTC. But remember we learned each one of us has 2 chromosome 7s. One from mom and one from dad. We have a homologous pair of chromosome 7s. A maternal homolog and a paternal homolog. So I'm going to represent that. So this is student 1, 2, and 3. They each have maternal chromosome 7. So I'll just put a number 7 here. Okay. Kind of move this up. And they each have a paternal chromosome 7. Okay. So let me quickly take a look at the data. Okay, yeah. Cool, awesome. So my restriction enzyme, or first let me back up. Let's say the gene taster gene here, this PTC tasting gene, is located up here. So we'll say PTC gene. Okay, so let's say the PTC gene is located right there, okay? So, like we learned with homologous chromosomes, they all have the same genes at the same location. I did mention, remember though, that they can be different versions of that gene. As we've seen in this lab, we have a taster version and a non-taster version. However, the same gene. They're both this tasting gene for this protein. They both have the gene, but they can be different versions of this gene, or alleles, as we've learned, as we've said. But they all have it at this location. This was the Heinz versus Hunt's ketchup analogy. Both ketchup, just different versions of ketchup. Hair color, right? Same gene for hair color, pigment, and some people have less pigment, some people have more, blonde versus brown hair. Okay. Now, depending on that version, what version that is, as we saw, if it's the taster version with that SNP that allows for the restriction enzyme, FNU4 something, H1 or whatever, if it's that version, the restriction enzyme recognizes that sequence and cuts there. It cuts the DNA. So I'm going to represent that with green here. And just so I know what the enzyme's called, FNU4H1 is our restriction enzyme. And I'm actually going to look at my data here for student 1 and 2 and 3, and accurately, I'm looking at the gels, show you where the restriction enzymes cut to determine, and then that can determine what their sequence is, the DNA, whether they're tasters or non-tasters. So let me see, student 1 looks like that. Student 2 is digested as that. Okay. So I'm just reminding myself. Okay, sweet. Student 1 is going to look like this. So FNU4H1, the restriction enzyme, is going to come in and cut either one. It could be that version, but this student has one taster version on one of their chromosomes, right? Remember, 2 alleles, 2 versions, because there's 2 genes here, right? And one of them could have been the maternal homolog, is the taster version. I'm going to show you why in a moment. For student 2, I believe student 2 has both as taster versions, meaning there's 2 cuts. Yes, okay. Like this. So FNU recognizes in both of the chromosomes, 2 cuts here. Close enough, H1. Student 3, however, both genes encode for the non-taster, meaning no cuts from FNU H1. Okay. So student 1, it cuts once. Student 2, twice. Student 3, no times. Let me show you what that would look like on a gel. So we amplify this gene with PCR. We apply the restriction enzyme. It cuts or not. Right, and this is after we've collected DNA from the individual, from their cheek cells. And we run the DNA that's cut or not on a gel, and that's our results. Let's look at student 1. There was one cut there. I'm going to wrap this up pretty soon because it's been about 45 minutes-ish. Okay, so student 1 has these 3 lanes. The ladder, by the way, is for reference. It's got a range of DNA sizes just to compare how large or small. They're known sizes. But let's take a look. So you have a control well with undigested DNA sample. So you don't put the restriction enzyme in. That's just a control to compare it to. So the digested DNA here in student 1, you have DNA at that's the same length, migrated the same distance, so it's same length as the undigested. That's this chromosome. There's no cut. This cut, however, produces a smaller DNA fragment and a little bit larger one. Okay? So if we look at the data, you can see that. There's a little bit larger one. Not from that one. So let me back up. See that very faint band down here? It migrated pretty far. That's our small fragment. That's our smallest one that I just showed. And then the one that's slightly smaller, just slightly smaller than the undigested one, slightly smaller than this one, is our bigger one. So you see how this can visually connect with our gel, our results here? So they have one chromosome that has a taster allele and one chromosome that does not. Okay, let's look at number 2. I'm going to wrap this up, I promise. Number 2, we have our control, our ladder. Here's our digested result. For number 2, we don't have a digested sample that's the same length as an undigested. So there can't be a chromosome that's undigested, or else there'd be a fragment at the same larger length. But we have 2 bands here, right? One that migrated a little further, so a bit smaller than the undigested, and one that migrated really far. Can you see how that correlates with this? 2 cuts. We've got these fragments here and these. Why aren't there 4 bands? Well, they migrated the same length. They're just on top of one another. So you can't really see it. They're the same size. But there's 2 cuts because there's no bigger giant undigested DNA fragment, because as you can see with the control in student 2, there isn't a same length DNA sample. This means that this student has 2 versions, 2 alleles of taster gene. And then finally, number 3, I'll kind of leave that to you guys to decipher, but it's the only other option. And I hope after seeing that, it makes sense. But I really like to walk through this lab because it's obviously a lot of understanding going on. And this whole dominant recessive alleles, it'll be even more clear on Thursday. Again, I apologize. I wish that kind of lined up better. But okay, that's it. That should give you everything you need to answer the pre-lab questions and the post-lab. And it's our last lab, so we're going out with a bang before your genetic research projects are due. But there's no more labs. Okay, that's it. So please email me if any of this doesn't make sense. If you have any questions, can set up an office hour. And yes, that's it. Okay, cool. How long was that? Could have been worse. All right. I'll see you guys Thursday. Asynchronous section, I won't see you, but let me know if you have questions.